Welcome to USC Ideas and Actions, the official University of Southern California podcast. Shows are webcast live on Facebook from Studio 1A in the USC Annenberg School for Communication and Journalism.
Hi, I'm Alex Capron. I teach at the USC Gould School of Law and the Keck School of Medicine of USC, where I teach health policy and bioethics. I've written about human cloning and stem cell issues, and one of my books is entitled Genetics, Ethics, and Human Values. Gage teaches a course called Stem Cells, Fact or Fiction, and I've given some lectures there on some of the ethical and legal issues that arise in stem cells. Today we're going to explore the possibilities and ethics around editing genes in stem cells and embryos. Stem cell researchers are creating chimeras. That is a mix of two different animals or even species. Can you talk about some of the chimeras that are common to stem cell research and how using them can move research from the labs to clinical applications for patients?
Yeah, so I think throughout history, people have had fascination with chimeric animals and a lot of mythology. So mermaids, kind of mixtures of maybe some sort of dolphin and human, and centaurs and minotaurs, other types of animals. And now, today we can mix stem cells together from two different species and make mixtures of animals. So we're, one of the visions for this is that we currently have a lot of people waiting for organ transplants, and we don't have enough human organs that we can give to those people, and a lot of people die because they don't get a satisfactory organ. And so one idea using chimera research is, can we grow animals like a pig with a human organ, like a human pancreas or a human heart, that's genetically matched to that person? So when that person needs a new heart or a pancreas, we can just harvest it from that farm animal and then rescue that person's life. And so already, we haven't done it between human and pig yet, but people have done this, for example, between rat and mouse. Mm-hmm. So they've made a rat, for example, where the entire pancreas in that rat is of mouse origin by putting mouse stem cells in that early rat embryo. And they've even gone farther where they can harvest then that mouse embryo from the rat and cure diabetes in that mouse by putting back a good mouse pancreas from that rat.
And in the case of transplants of a heart from a pig, we know that pig hearts are actually a pretty good substitute for a human heart.
Yeah.
So we could also have a situation where we're editing the genes in a way that would allow the human being not to reject that heart by removing certain triggers for rejection, certain factors, and also, of course, making sure that the pigs are free of viruses that are incorporated in their cells. Is that also part of that research?
Yeah, no, that's exciting. There's work from George Church, for example.
At Harvard?
At Harvard, who's gotten rid of all the retroviruses or all the transposons, in these mobile DNA elements in the pig genome, which are responsible for a lot of the immune rejection, the xenorejection. And so the idea is we can gene edit pigs so that when we grow human organs in them, those organs we harvest will be less rejection.
Right, yeah. Yes. So I guess one of the questions that comes up with that is, are we creating in a chimeric way something which isn't a human, or conversely, when we do it with an animal and are putting into a mouse, for example, if we put genes that are genes for the human brain, would we be creating something which we ought to treat like a human being?
Yeah.
And for the most part, I think we've been able to answer that by saying that's still so much in the future in terms of any significant qualities of the type that we look to say, well, you're a human being, and this is a mouse, and we can tell the difference right away. But there has been concern all along about any interventions that affect the brain or the nervous system, because that's kind of where we say we differ from other animals, is in our mental capacities and how we are self-conscious of ourselves as beings in ways that most other animals aren't, although there are some that do have that ability to differentiate and understand themselves and what they're doing.
Yeah. So one thing in chimeras is when you take cells from another species, say a rat, and put it in a mouse, if that rat makes an organ, because it's in a mouse, that organ looks like a mouse organ, not a rat organ where the cells are coming from.
Right.
So the idea is if you would put, say, human cells in a mouse, and those human cells would contribute to the brain, that would be a mouse-like brain.
Yeah.
So
in terms of its
yeah.
complexity and its parts, it wouldn't be a little tiny human brain in a mouse skull.
Yeah. But kind of related to that, another line of stem cell research are these organoids that people are making.
Well, you're doing some of that, aren't you?
Not directly, but in our institute. Yeah.
In the institute is making the kidney.
Kidney organoids, lung organoids, and actually we do have a brand new researcher joining next month, Georgia Quadrado, who's making human brain organoids in her lab.
And so an organoid is like a single cell version of a part of an organ?
Yeah, so what happens for organoids, what you do is you take human stem cells and you allow them to naturally grow and turn into brain tissue. And under the right conditions, you can keep these alive for up to a year. And they'll form all the different layers of the human brain in culture. And what Georgia Quadrado has shown is that those organoids are so complex, they can actually detect light. So they have a retina, and they can have the brain become active in response to light.
So that's the vision part of the brain that those organoids come from, and they're responding that way. And would the thought be that organoids drawn from different segments of the brain would each have the ability, or a simple version of the ability, to do whatever that part of the brain is doing?
Yeah, so that's one strategy. People are growing a particular part of the brain to study that.
And then how do we ever put those together?
But in her case, it's the entire brain is developing with all its different parts. So it's got the cortex, the part of your brain which does higher level processing. It's got the visual cortex that senses light. And so one question is, as we grow these bigger and bigger, will they start to have cognition?
Yes.
And that maybe that becomes
one of the issues that goes back, an ethical issue goes back a long time now, since the development of means of supporting human bodies through the intensive care unit, where the respiration is supported and drugs are given to keep blood pressure up and so forth, is at what point has a person died? And we usually measure death by the absence of circulation and respiration, but we came to recognize in the late 1950s and into the 60s that there were bodies that were existing because of these artificial interventions, where the brain had basically been destroyed because the brain cells had liquefied and they were no longer capable of doing anything. And we said, well, this should be a new way of defining death. But when you think about that, the idea was that, well, you're transplanting organs below the brain, the heart, the kidney, the liver, the lungs, or something that you've taken out of a body that's been determined to be dead. But if you flip it around the other way and say, but over here we have a brain, if we transplanted that brain that you've grown in your lab into this body, would that then be the person that it was before? And I think most people would say, no, it wouldn't be. It would look the same, and if you could make all the neurological and the vascular hookups so it was getting blood and it was living and was able to control the body, that brain would not be that person because it hadn't had that lifetime of experience. So part of what we think about with the human being is very particular, that it's their own lived experience and their relationships. That's what makes them who they are. We live in reaction to the world. But these new scientific developments are really going to be a big challenge to that. Now, when you think about this, one of the things that some animals have, but we don't have, is the ability to regenerate things that are damaged. I guess a salamander can grow a leg or something, other things like that. Why don't we have that ability yet? Because in a way, the stem cell research indicates that our cells are capable of
Yeah.
growing these parts. Why don't we just do that naturally and
Yeah.
and replace ourselves like Wolverine?
Yeah. Yeah, so that's kind of the million-dollar question. And the thing is, a lot of these other vertebrates, like lizards or even fish, are genetically very similar to us. 80 to 90% of the genes we have the same genes that they do.
Just like a zebrafish.
Yeah.
We are.
And so I think, for a while it was thought that mammals or, humans were much less regenerative than lizards and amphibians. But there's kind of a new idea emerging, and that's that really, right after we're born, we still have a lot of the regenerative potential that other. So recent studies have shown that a day-old mouse, you can cut away part of its heart and it'll regenerate its heart. Now by 7 days, after
Has
birth,
lost?
it can lost that. Whereas a zebrafish can do that throughout its entire life. So it's not really that we don't have the genetic programs to regenerate our tissues, but that for some reason we're genetically wired to lose those regenerative programs very early in life.
So there's some genetic signal that tells the cells not to do that.
Yeah.
And I suppose if we didn't have that and those cells were growing or potentially growing constantly, that could be something develop into a cancer or something.
Yeah.
I mean, you don't want your heart just growing and growing.
Yeah, so that's one idea, is that we shut down our stem cells in a lot of our organs to prevent cancerous growth.
Yeah.
But that's not the whole story. For example, in the heart, cardiac cancers are extremely rare. And so there's others ideas for why we don't regenerate. So for example, maybe there's certain costs that we take to regenerate, and we're putting that energy into other things such as reproduction or
Right.
other sorts of social structures.
Right. So a good deal of the research using stem cells is exploiting that ability that either embryos or blastocysts, pre-embryos, have with their stem cells to develop organs. And I suppose the earlier we go, the more different kinds of organs can develop from any particular stem cell.
Yeah.
One other thing, of course, that we've seen is the ability to take a cell, a so-called somatic cell in the body,
Yeah.
any cell, and create, reprogram it, put it into an embryo, and have the embryo develop with the genetic makeup of that cell we took. And most famously, of course, there was Dolly the sheep.
Yeah.
Now we have Barbara Streisand's dog that
Yeah.
she got cloned. Where is that research going to take us? What's useful about that capability of cloning?
Yeah. So cloning now is fairly widespread. So there's actually one famous researcher, Woo-Suk Hwang in Korea, who kind of came to notoriety in the mid-2000s, claiming to have cloned humans, at least done to make a cell line.
laboratory.
Yeah, to make a cell line through a cloning type of technology, and that was later found to be a fraud. But he is at the same time, he was actually the first one to clone a dog, and so nowadays he's kind of resurrected his career, and so he has a large company called Suam, which operates in Korea and China, and they're actually cloning several hundred dogs a year. And so one of the things they're doing is, for example, the military has these bomb sniffing dogs, which are very valuable, but very few dogs have this ability to really sniff out bombs well. So what they're doing is they're cloning these very valuable bomb sniffing dogs to see if that's a genetic trait, if they can create many more of these bomb sniffing dogs.
So is that a research question? Is it a genetic trait, or do they know that it has a genetic component and that?
Yeah. So for them, it's a practical thing. They want more of these dogs. But
Right.
it ends up being a research question, because they'll find the answer when they make
Right.
50, 100 of these dogs.
And if a
they'll-
lot of them
yeah.
can't sniff bombs particularly
Yeah.
well, it'll turn out it's some other characteristic that isn't just a genetically determined one, something in their
Yeah.
environment that.
And in this country, there's a lot of farm animals that are being cloned. And actually, I think it's legal to sell cloned meat from cloned livestock in the United States. Is that true? Are you aware of regulations on that?
That it's illegal? I'm not-
Legal, but there's no restrictions.
No, there's
on-
no restriction.
Yeah.
I wasn't aware. As you started to say that, I thought you said it's illegal.
No, I think there's
I don't
no
think
laws
so. No,
against it, right?
and there the characteristics that they're looking at are much more obvious. I mean, that certain animals put on the kind of, for example, meat on a bull or something that is what consumers want in their steaks.
Yeah, and I think a lot of the cloning is actually the stud animals.
Yes.
The valuable males that will...
Right.
Because it's not financially feasible to clone just a generic type of meat animal. You want a breeding animal that would be valuable.
But in terms of what would be done with human beings, why would we not want to clone a person, but what's valuable about the ability to get a cell to revert to a more pluripotent, if not omnipotent?
Yeah. So, there's the thing that makes the news a lot is, cloning human beings reproductively.
Right.
But there's also what's called therapeutic cloning. And that's the idea, let's say, if someone has, Alzheimer's disease, and you want to understand, well, why does that person have Alzheimer's disease? You want to study their neurons because Alzheimer's affects your brain. But you can't just go in and pluck out neurons from that person. But what you can do now is you can get a little bit of skin or even blood and add these factors, convert those cells into neurons. And so now you have neurons, and they're the neuron from the patient. They're genetically identical. And they may have genetic mutations that predispose that person to Alzheimer's. So now what you have are these neurons that are genetically identical from this Alzheimer's
and you can study
patient.
them?
And you can study them. You can find
Try
drugs that
will
correct them.
Yeah.
And now if you get a drug that corrects those patient-specific neurons in culture, that would be a personalized medicine for that person.
And with the new technique called CRISPR-Cas9, where we're able to do what's called gene editing and affect a particular genetic mutation, these cells, if you could also discover that the problem is caused by an identified mutation, go in and correct that. Would the idea then be that these cells, if they had a stem cell sort of activity, could go back in the brain and begin to create cells that would operate correctly as opposed to the patient's native cells? Or would the idea be that you would go in with the gene editing and try to affect all at once all the cells in the brain?
Yeah.
Is that too futuristic to know the answer?
I would say maybe in terms of the brain.
Yeah.
The brain is the most complicated organ, so
Right.
it's very difficult to imagine gene correcting in the billions of neurons. But say in the blood system.
Yeah.
So the blood system, there's only a handful of stem cells that make a whole new system of blood every week.
Mm-hmm.
So if we can have access to those blood stem cells, and we can go and use CRISPR to revert a mutation into a normal copy of a gene, we can completely restore someone's blood system to normal within just a week if we hit those stem cells.
And some people, that blood system is responsible for leukemia or a cancer or so forth.
Yeah.
And now what we have to do is find someone who's a very good match and transfer bone marrow cells, which are stem cells, to them after we've obliterated, through radiation, obliterated the patient's own cells and try to get these new transplanted cells. But this way you're describing, we would be able to go in and make a transfer of the patient's own corrected blood cells.
Yeah.
And you wouldn't have any of the other problems because sometimes after a bone marrow transplant, the patient has a reaction against that transplanted organ, the bone marrow, and rejects it. Well, all of these raise a lot of interesting ethical questions because while we think about these purely in therapeutic terms, if we were able to make an improvement, we could start off thinking, well, we're just going to improve people who have identifiable diseases. That is to say, someone has a problem in their genes that causes them not to be able to produce a protein that's necessary for life or that causes retardation or something. We're just fixing that. But if we begin to understand more about the effects of genes on human functioning, we can get to the point where we say, well, these are the genes that are associated, and there may be thousands of them that are interacting, but these are the ones that are associated with intelligence or strength or speed or disease resistance, your immune system, and so forth. And somebody could well say, well, wouldn't it be better if my kid got the corrections, not just to make them normal, but super normal.
Yeah.
And then we get to this whole question of designer babies and people saying at an embryonic stage or even at the in vitro stage where you just have an 8-cell being that has been created in a fertility lab, maybe even a couple that doesn't need fertility assistance to become pregnant, but says if it's outside the body in a lab, we can come in with these techniques and then we'll have a child that has more of the good kinds of traits that we want, and people will start thinking that's a better baby.
Yeah.
To me, that seems very problematic because one of the things we learn about life, particularly when we have children, is that there are a million ways in which our children are going to develop whatever their genes are. And there's the chances of life and of circumstances. And if we're doing any of this because we want a particular child, we're likely to be disappointed.
Yeah.
And if our acceptance of the child and our support for the child and our love for the child is dependent upon that child fulfilling our image of what the good child should be, and they disappoint us, the disappointment will go beyond that. I mean, every parent has some fantasy of their child having a wonderful life and so forth, but we all come to realize that there's a lot of chance with that, and what's wonderful gets really defined by the child more than it gets defined by us. And so I see that as an issue about the ethics of life and of families, where something that's being developed for very good reasons to cure disease is going to create a capacity that could be used in ways which are not so good.
Yeah. So I think like a good example of that is work being done on HIV right now. So, for example, Paula Cannon, who's at the Keck School, she's been studying that there's a particular gene that if someone's blood cells don't have this gene called CCR7, their T cells cannot be infected by HIV. So these are completely HIV-resistant people.
Right.
And that's actually a cure. One cure for someone who has HIV is to knock out this gene in their blood system. So now that their cells can no longer be infected by HIV and they can clear it.
Yeah.
But then you get to these issues at a population level. What if you're, I think we're getting HIV somewhat under control now, but what if it was still exploding out of control? Would it make sense to edit human embryos so that they don't have this receptor?
They would be born immune to.
And we know that there's 10% of the population out there that don't have this gene, and they seem to be fine.
Yeah.
But then the question is, how do we know that
right.
there's not some negative consequence of
right.
losing this gene?
Well, I mean, certainly we have found a lot that ought to make us very humble when we start saying we know this does this or that.
Yeah.
Because the description at the beginning of the Human Genome Project, which was, I guess, roughly about 30 years ago, that once we had this blueprint, we would understand human beings.
Yeah.
And we've come to say, well, wait a second, it's not just the DNA, it's the RNA, it's the expression of genes, it's the whole, now we call proteomics, it's understanding the proteins that are made. And if we had rushed ahead, if we'd had this ability and rushed ahead, and then it turned out that having that gene has some other controlling consequence in the human being that could be adverse, and now we've been editing all the embryos, then that whole generation is then at risk for whatever problems. And we really need to have some humility as we declare our ability to remake human beings.
Yeah. But is it somewhat inevitable? It seems like society as a whole, the technology is there. Like IVF, for example, was very controversial in the 70s and 80s, right? And now it's largely accepted in society.
It is accepted, but there are data that indicate that IVF embryos, when they grow up, have a slightly higher range of medical risks on a number of factors.
Yeah.
And so if you looked immediately after IVF was developed, we were looking for the sort of the grossest kinds of abnormalities, and those were not occurring at higher rates. I mean, there were, as with normal pregnancies or normal reproduction, there was fetal loss. That is to say, people had spontaneous miscarriages and so forth. We have a question coming in from Facebook. What can stem cells do for osteoporosis?
Okay. You want me to take that?
Well, I certainly can.
Yeah, so osteoporosis is a condition most commonly in postmenopausal women, and so essentially your bones are very dynamic organs. You're constantly adding bone and taking bone away, and what happens is that over time, especially after menopause, you have too many of the cells that take away bone, and not enough of the cells that make bone. And so I'm not sure if this is a treatment that's really primed for stem cell type of therapy, because there's a lot of drugs that are being developed to kind of alter this balance to increase the cells that make bone and prevent the cells that are taking away bone to kind of restore that healthy balance of bone. But I suppose you could imagine if we lost these bone making cells, we could make bone making cells in culture and then transplant them into the patient. So there's a potential there.
And those are the kinds of cells we would expect to be able to take up some space and grow and fasten on. I mean, they're not.
Yeah,
they're not
and so we know
cells.
there's stem cells, for example, in the marrow of their bone.
Yeah.
That fatty, when you break open bone, that fatty.
Right.
But there's a lot of stem cells in there that are creating these bone making cells. So it may be that as we age, we lose those bone stem cells. And now that we know a lot about how to make these bone stem cells, we could imagine making them even patient specific and giving them back to the patient. Okay, so we have another question from Facebook Live. So, a rabbi in Israel claimed that cloned pig would be kosher.
I guess that's cloned as a source for an organ transplant.
Yeah, but okay.
Oh, is he saying? Well, I don't know. Cloned pig would be okay for dinner.
Okay.
Because it would.
Oh, does cloning change the genetic makeup that much? It'd be kosher. So really, cloning it'd still be a pig. Right?
Yeah.
The genome is what determines it's a pig. It's
right.
Exactly.
Cloning doesn't change that. Cloning aims just the opposite. It intends to keep the same genome in the offspring as in the progenitor, as it were. But gene editing is really something that could change the genetic makeup, and that's what people like Professor Church are using on the pigs that they're trying to make acceptable potentially as donors. I think they've actually used them with a primate. I think he has taken a heart and may have put it in a non-human primate, I believe. But in any case, I think that's where they're headed for.
Yeah.
But that's not cloning. That's gene editing to take out the retroviruses that are.
I just say like a follow-up on that is that there's a very interesting. A lot of companies have started to make in vitro meat, where you can take muscle stem cells from a cow, from a chicken, or a pig, and make meat in vitro using these muscle stem cells to make unlimited amounts of pig muscle. So that'd be an interesting question whether that's kosher, right? The in vitro pork.
Yeah, and that I guess is the cells are cloned, so maybe that's what he was referring to. The idea is that that's attractive to people who don't want to kill animals in order to create a food supply, and who also notice the harmful effects of animals on the environment when they're raised in large numbers as a human source of meat.
Yeah. So the next question is, can stem cells be used to produce new teeth? It's fascinating. Many of us lose our teeth, and we need artificial implants, and those teeth are, they work pretty well, but they're not like natural teeth. And so I think there's a lot of good research going on. The tooth is actually fairly complicated because you have different parts of the tooth made from different types of tissue. So you have your enamel on the outside, the dentine, but you also have these roots that need to go into the bone and attach. And in particular, we can make the outside of the tooth, the enamel and dentine, but making those roots that allow it to attach is what's been really tricky so far. So we'll probably get there. I don't know if it'll be 20 years or 50 years, but I think that is something that will be tractable in the future.
One of the other things that comes up with all of these things which are aimed to be regenerative and to stop and really reverse natural aging is what would happen if we were enormously successful with those and people were not aging and dying. And, that search for the fountain of youth has motivated people for centuries.
Yeah.
It would be a very different society if we didn't have normal generational turnover. There are many ways in which it's not simply the ability to do work and to be strong and not to be burdened by old age diseases, but there is something about new people with new experiences that brings something very valuable to society. And the notion that we would kind of freeze our population at any time and not have new people coming along. We already have an overpopulated world. It would be very odd to say, well, we're going to keep everybody who is alive now alive and still go on having babies. And the ability to have families and so forth has been a very important part of the structure of society. So I would be very dubious that there is a good reason to try to keep people alive indefinitely or to regenerate all their organs so they can live indefinitely.
Yeah. Okay, so...
well, maybe we should answer this question. When will stem cells help treat people with diabetes?
Yeah, I think we're almost there. People like Doug Melton have been able to take human embryonic stem cells and make them into insulin-secreting cells, which a lot of people with diabetes just don't have enough of these insulin-making cells. And there's clinical trials going on right now implanting them, these stem cell-derived insulin-producing cells to cure diabetes, and it's looking very good in animal models. The one issue is, how do you put these cells back into a person and not have them die off within a couple of months? So it's going to work, but for how long will it work is the question right now.
And I suppose that's a particular issue with type 1 diabetes, where the reaction of the body against those very cells, that if you create more of them, you have a short-term reaction, and then whatever is wrong with the immune response that is treating these cells as foreign cells, which is what happens, would go on with those new cells, and you'd be no better off.
So I think what researchers are trying to do is implant them in devices that very
that
small
protect
pores, so the immune cells can't get in and attack those cells. But insulin can still get in and out, and glucose can get in and out. So they can know how much glucose is in the blood and produce the insulin, but they're protected. It's like an armor with these little tiny holes.
Right.
And so there's several companies that have been working on these devices now. So will we be curing arthritis with stem cells? I think maybe one day. I think that we're still a long ways off. We know very little about stem cells that can generate new cartilage at our joints. And so I think we're in very early days. So as opposed to diabetes, which I think is going to be something we'll see in the next few years, I would say arthritis is maybe 20, 30 years off, if I had to guess.
Well, I think we've come to the end of our show, and it's time to thank the Annenberg Studio A for letting us broadcast today. I'm Alex Capron from USC Gould School of Law and Keck School of Medicine.
And I'm Gage Crump from the Broad Stem Cell Center at the Keck School of Medicine. And so thanks for joining us.
Thanks for listening. Subscribe to our podcast and follow us on Facebook.